Hello everyone. In today's biology MCQ session, we are discussing important questions from the chapter Biotechnology Applications. Question number 1. The first genetically engineered human insulin produced commercially is known as Actrapid, Humulin, Novolin, Insugen. The right answer is option B. Humulin was the first recombinant human insulin produced using genetically engineered E. coli. It was developed by Genentech and marketed by Eli Lilly Company. Question number 2. Which of the following is used as a selectable marker in recombinant DNA technology? Ampicillin resistance gene, lactose gene, insulin gene, trypsin gene. The right answer is option A, ampicillin resistance gene. Selectable markers such as ampicillin resistance gene allow identification of transformants by providing antibiotic resistance. Only recombinant bacteria survive in antibiotic medium. Question number 3. The bacterium used to produce Bt toxin in Bt cotton is dash. Bacillus subtilis, Bacillus anthracis, Bacillus thuringiensis, Bacillus megaterium. The right answer is option C. Bacillus thuringiensis produces cry proteins toxic to certain insect larvae. Bt genes are transferred into plants to provide insect resistance. Question number 4. Assertion. Bt toxin is produced as an inactive protoxin in bacteria. Reason. The toxin becomes active only in the alkaline gut of insects. Both assertion and reason true. Reason explains assertion. Both assertion and reason true, but reason not explanation. Assertion true, reason false. Assertion false, reason true. The right answer is option A. Both assertion and reason true. Reason explains assertion. Bt toxin is initially an inactive protoxin. In the alkaline midgut of insects, it gets activated and destroys gut cells, causing death. Question number 5. Golden rice is genetically modified to contain high amounts of vitamin C, iron, lysine, beta-carotene. The right answer is option D, beta-carotene. Golden rice is enriched with beta-carotene, a precursor of vitamin A. It helps to prevent vitamin A deficiency, especially in developing countries. Question number 6. Which of the following crops is genetically modified for pest resistance using Bt gene? Rice, cotton, wheat, barley. The right answer is option B, cotton. Bt cotton contains cry genes that produce toxin harmful to bollworms. It reduces pesticide use and increases crop yield. Question number 7. The term gene therapy refers to replacement of defective genes, treatment using antibiotics, artificial fertilization, vaccination. The right answer is option A, replacement of defective genes. Gene therapy involves introducing normal genes into cells to correct genetic disorders. It is mainly used for inherited diseases. Question number 8. The first successful clinical gene therapy was given for hemophilia, cystic fibrosis, ADA deficiency, sickle cell anemia. The right answer is option C, ADA deficiency. The first gene therapy was performed in 1990 for adenosine deaminase deficiency, a severe immune disorder. Question number 9. Which vector is commonly used in gene therapy? Protozoa, algae, fungi, virus. The right answer is option D, virus. Modified viruses act as vectors to transfer therapeutic genes into host cells because of their ability to infect cells efficiently. Question number 10. Statement 1. Cry genes are named according to the insect they affect. Statement 2. All cry proteins are toxic to the same group of insects. Both correct, only one correct, only two correct, both incorrect. The right answer is option B, only one correct. Statement 1 is correct because cry genes are named based on their target insect. Statement 2 is false because different cry proteins target different insect groups, not the same group. Question number 11. Match the following. Column 1. ADA deficiency, Bt cotton, golden rice, recombinant insulin. Column 2. Vitamin A enrichment, diabetes treatment, gene therapy, cry gene. The right answer is option D, A3, ADA deficiency gene therapy, B4, Bt cotton cry gene, C1, golden rice vitamin A enrichment, and D2, recombinant insulin diabetes treatment. Question number 12. Which enzyme is responsible for cutting DNA at specific sites? Restriction endonuclease, DNA ligase, DNA polymerase, reverse transcriptase. The right answer is option A. Restriction endonuclease recognizes specific palindromic sequences and cut DNA producing sticky or blunt ends. Question number 13. RNA interference helps in controlling pest by dash gene expression, increasing, activating, silencing, duplicating. The right answer is option C, silencing. RNA interference causes gene silencing by degrading mRNA. This prevents protein synthesis of harmful genes in pest. Question number 14. Which organism is used in producing recombinant insulin? Saccharomyces cerevisiae, Escherichia coli, Bacillus subtilis, Agrobacterium tumefaciens. The right answer is option B. Escherichia coli is used to produce recombinant insulin because it grows rapidly and can be genetically manipulated easily. Question number 15. Assertion. Genetically modified crops reduce the need for chemical pesticides. Reason. They possess built-in resistance to pest. Both true and reason explains assertion. Both true but reason not explanation. Assertion true, reason false. Assertion false, reason true. The right answer is option A, both true and reason explains assertion. Genetically modified crops like Bt cotton produce insecticidal proteins themselves, reducing reliance on external pesticide application. Question number 16. Biopiracy refers to hybridization, artificial cloning, mutation breeding, illegal use of biological resources. The right answer is option D, illegal use of biological resources. Biopiracy involves unauthorized use or patenting of biological resources or traditional knowledge from a country. Question number 17. Which of the following is an example of biopiracy? Neem pesticide patent controversy, Bt cotton production, golden rice development, tissue culture. The right answer is option A, neem pesticide patent controversy. Patents on neem products by foreign companies were challenged because neem had long been used traditionally in India. Question number 18. Which molecule triggers RNA interference? rRNA, tRNA, double-stranded RNA, mRNA. The right answer is option C. Double-stranded RNA activates RNA interference, which degrades complementary mRNA molecules. Question number 19. Which of the following is a transgenic animal? Hybrid maize, sheep producing human protein in milk, high-yield wheat, artificial inseminated cow. The right answer is option B, sheep producing human protein in milk. Transgenic animals carry foreign genes introduced using biotechnology and can produce valuable proteins. Question number 20. The most common transgenic animals used in research are rabbits, rats, mice, sheep. The right answer is option C. Mice are widely used because they reproduce quickly and their genetics is well understood. Question number 21. The technique used to detect specific DNA sequences is called dash, southern blotting, northern blotting, western blotting, ELISA. The right answer is option A. Southern blotting identifies specific DNA fragments using complementary probes. Question number 22. Which test is commonly used for detection of HIV infection? PCR, ELISA, western blot, southern blot. The right answer is option B. ELISA detects antibodies against HIV antigens and is widely used for initial screening. Question number 23. Statement 1. PCR amplifies DNA sequences. Statement 2. It requires DNA polymerase enzyme. Both correct, only one correct, only two correct, both incorrect. The right answer is option A, both correct. Polymerase chain reaction amplifies DNA using DNA polymerase, primers, and thermal cycling. Question number 24. Which of the following is not a purpose of transgenic animals? Studying gene regulation, producing vaccines, testing chemical safety, increasing natural mutation rate. The right answer is option D, increasing natural mutation rate. Transgenic animals are used for research, vaccine production, and toxicity testing, not for increasing mutation rate. Question number 25. The method used to introduce foreign DNA directly into plant cells using high-velocity microprojectiles is called dash. Electroporation, gene gun method, microinjection, liposome-mediated transfer. The right answer is option B, gene gun method. The gene gun method delivers DNA coated on microscopic gold or tungsten particles into plant cells. It is widely used for transformation of plant tissues that are difficult to infect with agrobacterium. Question number 26. The TI plasmid used in plant transformation comes from Bacillus thuringiensis, Rhizobium leguminosarum, Agrobacterium tumefaciens, Azotobacter. The right answer is option C, Agrobacterium tumefaciens. Agrobacterium tumefaciens transfers tDNA from its TI plasmid into plant cells, making it useful for plant genetic engineering. Question number 27. Assertion. Transgenic animals are mainly used to increase agricultural yield. Reason. Transgenic animals help in testing vaccine safety before use in humans. Both assertion and reason true, and reason explains assertion. Both assertion and reason true, but reason not explanation. Assertion true, reason false. Assertion false, reason true. The right answer is option D, assertion false, reason true. Transgenic animals are primarily developed for research, disease studies, and production of biological products, not for increasing agricultural yield, but they are used in testing vaccine safety and studying immune responses. Therefore, assertion is false, reason is true. Question number 28. Which of the following is used to produce insulin chains A and B separately? Yeast, bacteria, virus, algae. The right answer is option B, bacteria. In earlier recombinant insulin production, E. coli bacteria produced separate A and B chains which were later combined. Question number 29. Bt toxin forms dash in the gut cells of insects: protein pores, enzyme complexes, DNA fragments, lipid droplets. The right answer is option A, protein pores. Activated Bt toxin bind to the gut epithelial cells and form pores, causing cell lysis and insect death. Question number 30. Which of the following best describes biotechnology applications in medicine? Production of antibiotics only, genetic diagnosis and gene therapy, mutation breeding, selective breeding. The right answer is option B, genetic diagnosis and gene therapy. Biotechnology in medicine involves recombinant drugs, gene therapy, PCR diagnostics, vaccines, and genetic testing. Question number 31. Which of the following statements about cryproteins is correct? They are toxic to all insects. They are produced by viruses. Different cryproteins target different insect groups. They destroy plant cells. The right answer is option C. Different cryproteins target different insect groups. Cryproteins produced by Bacillus thuringiensis are highly specific. Different cryproteins such as Cry1, Cry2, Cry3 target different insect groups. Question number 32. The genetically engineered rice enriched with provitamin A is called dash: silver rice, hybrid rice, BT rice, golden rice. The right answer is option D, golden rice. Golden rice contains genes for beta-carotene synthesis in the endosperm. It prevents vitamin A deficiency. Question number 33. Assertion. RNA interference is used to develop pest-resistant plants. Reason. It prevents translation of specific mRNA in pest. Both assertion and reason true, and reason explains assertion. Both true but reason not explanation. Assertion true, reason false. Assertion false, reason true. The right answer is option A. Both assertion and reason true, and reason explains assertion. RNA interference causes gene silencing by degrading mRNA molecules. This prevents synthesis of essential proteins in pest. Question number 34. Which of the following genetically modified crops was developed to reduce post-harvest losses by delaying fruit ripening? BT cotton, Roundup Ready soybean, golden rice, flavor sour tomato. The right answer is option D. Flavor sour tomato was the first genetically engineered food approved for human consumption. It was modified to slow down the softening process, thereby delaying ripening and increasing shelf life. Question number 35. Which of the following techniques is used for early detection of pathogens? Tissue culture, PCR, hybridization, fermentation. The right answer is option B, PCR. Polymerase chain reaction amplifies pathogen DNA. It allows detection even when pathogen numbers are extremely low. Question number 36. Statement 1. Transgenic animals are useful for studying gene regulation. Statement 2. They contain genes introduced from another species. Both correct, only one correct, only two correct, both incorrect. The right answer is option A, both correct. Transgenic animals carry foreign genes inserted artificially. They help scientists study gene expression and regulation. Question number 37. Which of the following is an example of a recombinant therapeutic protein produced using biotechnology? Penicillin, human insulin, ethanol, citric acid. The right answer is option B, human insulin. Recombinant human insulin is produced by inserting human insulin gene into bacteria like Escherichia coli. These bacteria then synthesize insulin, which is purified for the treatment of diabetes. Question number 38. Which component of Bt toxin causes cell lysis in insect gut? Lipid enzyme, DNA fragment, protein crystal toxin, carbohydrate polymer. The right answer is option C, protein crystal toxin. Bt toxin exists as protein crystals. After activation in the insect gut, they create pores in the epithelial cells, causing cell lysis and death. Question number 39. Which organization in India regulates the safety of genetically modified organisms? WHO, GEAC, FAO, ICMR. The right answer is option B, GEAC. Genetic Engineering Appraisal Committee under the Indian government approves the environmental release of genetically modified organisms. Question number 40. Transgenic animals producing useful proteins in their milk is known as dash. Biopiracy, tissue engineering, gene cloning, molecular farming. The right answer is option D. Molecular farming involves producing pharmaceuticals like human protein in the milk of transgenic animals. Thank you for watching the video. Subscribe to the channel to learn more biology MCQs.